Hello everyone, the next topic which we will be discussing is about the flagella. So what are flagella? Flagella, they are long thread-like cell organelles that are present on the surface of a living cell. It is mainly found in the prokaryotic as well as in the eukaryotic cells. So the word flagellum, the flagella is a word, but the word flagellum which means whip. And these hair-like structures, they are involved in the locomotion of the cell. So let's start with the first slide, that the word flagellum, which means the whip. Here is a diagrammatic representation of the cell where they are representing these hair-like extensions as the flagella. So what these hair-like extensions do? These hair-like extension will help in the movement of the cell. And there are some special flagella that are used in few organisms as the sensory organs, and that will sense the change in the pH and the temperature. Now, there are three domains of life of the flagella. They can be bacterial flagella, they can be archaeflagella, and they can be eukaryotic flagella. So what are these? First we will discuss about is the bacterial flagella. They are a coiled thread-like structure, sharp bend, which consisting of a rotatory motor at its base, and they are composed of the protein flagellin. Next is about the archaeflagella. They are non-homologous, so they are non-homologous in nature. Then eukaryotic flagella, they are complicated cellular projections that pumple backwards and forward and are found in the protist cells, gametes of the plants and the animals, and it is made up of a protein which is called as the tubulin. So it is made up of which protein? It is made up of the protein which is known as the tubulin. Now we will be discussing the bacterial flagella in detail. So the flagella is a helical structure which is composed of the flagellin protein. So now this bacterial flagella, they are being divided into three parts further: basal body, hook, and the filament. Now if we discuss about the basal body, it is attached to the cell membrane and the cytoplasmic membrane. It consists of rings which is surrounded by a pair of protein that is known as the MOTB. So it consists of a protein which is known as MOTB. And these rings, the basal rings, includes the L ring, P ring, and the C ring and the MS ring. So they are composed of these types of rings. Now if we talk about the L ring, they are outer ring anchored in the lipopolysaccharides layer, and they are found in the gram-positive bacteria. So they are found in the gram-positive bacteria. If we talk about P ring is anchored in the peptidoglycan layer. C ring, it's for anchored in the cytoplasm, and MS ring, it is anchored in the cytoplasmic membrane. So this is the basal body, and these are the four rings that are being depicted. Now if we talk about the next structure of the bacterial flagella, that is hook. It is a broader area which is present at the base of the filament. So they are present at the base of the filament. They connect filament to the motor protein in the base, and the hook length is greater in gram positive bacteria. So they are present in greater in the gram positive bacteria. Next is the filament. So they are thin hair-like structures that is arising from the hook. So they are thin hair-like structures that is arising from the hook. Next is there are four types of flagella. So these are the four types of flagella. So first is periphery ocus, that is there are several flagella that are attached all over the organism. So they are attached all over the organism. and they are not polar flagella because they are found over the organisms. These flagella, they are rota anti-clockwise, and they form a bundle that will move the organism in one direction. So they will move the organism only in the one direction. So if some of the flagella break and they start rotating clockwise, the organism does not move in any direction, and they begin with the tumbling. Next is monotriasis. It is a mono, which means there is a single flagellum at end or the other. So these are known as the polar flagellum. So we discussed this. This was the non-polar flagella, but monotriasis is the polar flagellum, and they can rotate clockwise as well as in the anti-clockwise direction. So they will rotate in both the directions. And the clockwise movement moves the organism forward, while the anti-clockwise moves it towards the backward. So this is the two. Then the lopotriasis, that is, there are several flagella which is at one end of the organism or the other, and these are known as polar flagellum, and they can rotate clockwise as well as in the anti-clockwise. The clockwise movement moves the organism forward, while the anti-clockwise move it towards the backward. Then the ambiphytriasis, that is a single flagellum that is on both the ends of the organisms. They are known as polar, and they can rotate in both the directions, that is in the clockwise as well as in the anti-clockwise direction. And the clockwise movement moves the organism to the forward direction, and the anti-clockwise moves to the backward direction. So clockwise will move it to the forward, and the anti-clockwise will move it to the backward of the backwards. Next is about the functions of the flagella. So flagella, they help in the movement, organisms in the movement. They act as sensory organs to detect the temperature and the pH changes. It was being depicted in the first slide itself, that they will have a sensory organs that will detect the temperature and pH. Then there are few eukaryotes that use flagellum to increase the reproduction rates. Now there are some recent research that have proved that flagella are also used as a secretory organelle. So this is the example. Now the difference between the cilia and the flagella. So what are the main difference between the two? So the main difference is, first we will go with the definition of the cilia and then with the flagella. So definition is, cilia, they are short hair-like organelles that are extending from the surface of a living cell, whereas flagella, they are long thread-like cell organelles that are present on the surface of a living cell. If we talk about cilia, it is found only in the eukaryotic cells. But in case of flagella, they are found in the prokaryotic as well as in the eukaryotic cells. So they are present in both the cells. Cilia, they are generally short and hair-like organelles, and flagella, they are a long vipe or like organelles. So as you can see in the figure as well, the flagella, they are long in length, whereas cilia, they are short and they are very attached to the surface of the eukaryotic cell. Next, cilia is present throughout the cell, surface of the cell, whereas flagella, it is present at both the ends all over the surface. So it is only present at the ends of the surface, and it is present all over the surface. So through the figure also we can say the difference, or we can detect the difference between the two. If we talk about the density, cilia, it is present in almost in many, like in hundreds per cell, but in case of flagella, it is present in few per cell. So they are present in few. The cilia, they helps in the motion of cilia. The motion of cilia is rotational and very fast moving, so they move very fast. But in case of flagella, they have a rotatory movement in prokaryotes, while it is in the bending movement in the case of eukaryotes. So both the movements are different in case of the cilia and as well as the flagella. cilia beat in coordination or one after the other, but flagella beat independent of each other. The main functions of cilia includes it helps in the locomotion, aeration, feeding and circulation, whereas the flagella only helps in the locomotion. So paramecium is the example of cilia and salomina is the example of flagella. So this was all about the difference between the cilia and the flagella. Now these are some of the references. Thank you.